Chapter 1: Introduction to Protein-Based Drug Development. Protein-based therapeutic drugs, such as monoclonal antibodies, are typically available as injections and infusions. But how are safe and effective injectable therapeutic proteins made? What tools are needed to ensure that a stable therapeutic protein is injected into a patient? The manufacture of protein-based therapeutic drugs requires a multi-step drug development process. This process involves collaboration between individuals from multiple areas of expertise, including drug substance, drug product, attribute sciences, devices, manufacturing, legal, clinical, and patients. Altogether, the drug development process from initial research to being approved for patient use can take a decade or more. Chapter 2: What is process development? The drug development process has many stages. The early phase of drug development is dedicated to identifying the lead, or the ideal, drug candidate from a collection of potential candidates. Following this, the drug development process is divided into two parallel pathways: process development and clinical development. As its name implies, the process development pathway involves the development of a process for making the drug candidate on a large scale. In the clinical developmental pathway, the drug candidate is evaluated for its safety and efficacy, and how it is metabolized in the body. Process development can be divided into three categories: drug substance process development, drug product process development, and attribute sciences. In drug substance process development, scientists produce the therapeutic protein in living systems and purify the therapeutic protein to remove impurities. Scientists must perform analytical tests throughout the process to evaluate the purity of the drug substance, the term that's used to refer to the active molecule that's responsible for a drug's therapeutic effect. In drug product process development, the final formulation of a drug substance is stored in its designated container. For a protein-based therapeutic drug, this is either a vial or a syringe. Various assays are performed throughout the process to ensure the stability of the drug product for the intended storage time. In attribute sciences, scientists develop specific methods for monitoring a drug product's quality attributes. These are measurable properties or characteristics of a drug that help to define its overall quality. Various analytical assays are performed throughout the manufacturing process to understand the attribute impact on the safety and efficacy of the purified drug substance and drug product. Chapter 3: What is drug product process development? In this pathway, we'll focus on the key steps that are involved in drug product process development. In particular, we'll take a closer look at how a drug substance is formulated and stored in its designated container to create a drug product. To begin, we'll consider the form in which a drug product is stored. Depending on the indication, or the use of a given drug for treating a particular disease, the patient population, and the stability of the protein, a drug product can be stored in a liquid or in a lyophilized or powdered form. First, we'll learn how scientists decide between a liquid or a lyophilized version of the drug product. Next, we'll examine protein stability. In order to maintain its safety and efficacy, a therapeutic protein must remain in its native, or properly folded and functional, structure. Various stress conditions during the manufacturing process can challenge protein stability. A loss of its native structure can lead to an unstable protein, which results in a drug product that's not safe to inject into a patient. We'll examine the analytical tests and procedures that are performed to ensure the stability of a protein-based drug product. Once a drug product has been stabilized, it can be manufactured on a large scale and filled into its designated container for patient use. A common test that's performed at the filling stage is the visual inspection of the final drug product. We'll learn how manufacturers use the visual inspection process to ensure that the drug product is free from any unwanted materials, which could reduce its safety and efficacy. After filling, a drug product is ready to be shipped out for patient use. Protein-based drug products are typically administered to patients as an injection or an infusion. The final route of administration to the patient is determined by factors such as disease indication, dosage size, that is, the amount of medication taken at one time, and viscosity of the drug product. To learn more about how a drug product is given to patients, we'll look at the different routes of administration that are typically used for a therapeutic protein and the points of consideration for selecting each route. Lastly, to increase patient convenience, scientists can collaborate with engineers to develop a combination product. A combination product is a drug product that's combined with a device for easier drug delivery. We'll review how devices are used and selected based on patient input to ensure a convenient drug administration option for patients. Chapter 4. Conclusions. To recap, drug product process development is an important part of the overall drug development process. The numerous steps in this complex process collectively help drug manufacturers to deliver drug products that are safe, effective, and convenient for patient administration.